I would like to introduce our webinar speaker, Dr. David Han. David is Associate Professor of Cell Biology at the University of Connecticut Health Center. He is interested in utilizing proteomics and systems biology approaches to identify cancer biomarkers. His group has developed a method termed Genomics-Driven Immunoproteomics, or GDI, which utilizes high-density peptide microarrays to identify breast cancer-specific tumor antigens and antibodies that recognize these antigens. The eventual goal is to develop a multi-targeted anti-cancer therapeutics using the GDI approach. So the title of the presentation is Identification of Personalized Cancer Antigens and Antibodies Against These Antigens Using PEPperPRINT Peptide Microarrays. So once again, please use the question pane to type your questions, and we will also share the slides and the post-webinar file. So David, I'm handing the cursor over to you. Please take it away.
Thank you, Custom, and thank you everyone for signing in. Yes, as Custom has nicely introduced, our lab has been interested in studying cancer and a number of other diseases using proteomics and system biology tools. And the eventual goal, of course, is to provide usable therapeutics or diagnostics for the patients. Ideally, our research will end up in clinical settings. So what I'd like to do today is first talk about a tiny bit of historical aspect of how we started using PEPperPRINT microarrays and why we think it is very useful. So first, I'd like to start by talking about what we started doing about probably 10 years ago, what we call direct tissue proteomics. Essentially, at that time, there's a debate of how best to find cancer biomarkers. And one camp said, well, similar to prostate-specific antigen, which you can find in the blood, you should look for serum biomarkers. Whereas the other group thought that, well, if you want to find specific biomarkers, you should go to the source tissue and profile it. And we are in the second camp. So we started doing protein profiling, comparing cancer from the tissues versus normal. For example, if you look at this slide, you know, these are breast cancer tissue. These are stained with H&E. And you can see the cancer and surrounding stroma matrix, as well as a lot of lymphoid follicles that invade and try to attack the tumors. And those we now appreciating more and more of why these are very important for immunity. Essentially, what we did was look for somewhat homogeneous cancer cell rich region and core out these samples. So these are using biopsy needles, and then we select them. We lyse these tumor cells, and then we would run on a gel, and we cut gel into slices and identify the proteins. So this paper was published comparing estrogen receptor positive and negative breast cancers, and we identified approximately close to 3,000 proteins, and we were able to identify 150 upregulated, 90 downregulated proteins. Again, those were very useful, but one of the key points that arise from this kind of comparative studies is that can we find biomarkers that are truly specific to cancer cells? And if so, what do they look like? So one of the ideas then become, can we identify mutations that are present in cancer cells directly using proteomics? Well, at that time, typically the way to identify proteins is you would cut the gel bands, digest with trypsin, shoot in the mass spectrometer, and compare the peptide spectrum against normal human protein database. So obviously all the mutations are not part of the database. So then you would miss them because the masses would be different. So then we started making mutant-specific databases. So the question becomes, what kind of mutations do you download? So we started downloading from available mutation data from publicly available databases. And then we were just comparing our samples against these mutations that's been commonly described in the database, which I think is not necessarily very systematic, but that's how we started. As you can see in the next slide, here's an example of a mass spectrum. On the top is the wild-type peptide of a translational initiator protein, where you see wild-type spectrum, and in the bottom is the mutant spectrum. So the third amino acid, MFV valine, is changed to MFL leucine. So as a result, you have 14 Dalton of mass shift. So which you can see here, MFV B ion is 378, whereas MFL is 392. So then we showed that we can identify a number of mutant proteins, not just point mutations. If you were to look at insertions and deletions, for example, this is a very famous gene, CDKN2A, which is a cell cycle regulator. This is a 21 nucleotide insertion that occurs. And as a result, you get this wild type peptide sequence is changed into mutant peptide sequence. And we were able to identify these mutations as well. So the first step was, well, yes, we can profile and we can identify mutations directly in tumor tissues. Now, these mutations are expected to be present only in the cancer cells. So then it becomes logical that we should be using those as targets to either diagnose or develop therapeutic strategies to target these tumors. So how do we do that? Well, what then becomes is a PEPperPRINT Peptide Microarray comes in. So around that time, one of the key questions that was unclear was, well, if you were to look at the tumor tissue and you see hundreds or sometimes thousands of mutations, are these mutations actually immunogenic? Meaning, our immune system has developed ways to distinguish self and non-self, whereas the immune system is trained to recognize self, but non-self proteins are not recognized. So then if a tumor cell is making hundreds or thousands of mutant proteins, shouldn't they be recognized by the immune system? And if so, is there a rapid way to screen for it and identify it? Because the way we understand it now, most of the intracellular proteins that are made are degraded into proteosomes and loaded onto HLA peptides and displayed on the cell surface. The key point there is not all peptides that are degraded can fit into HLA binding group. So for example, a protein with 1,000 amino acids may present 1 or 2 peptides that can fit into HLA binding. So the rest of the peptides would not be displayed. So then the question is, among all the hundreds of thousands of mutant proteins, which one of those can actually fit into the HLA peptide binding groove? And if so, can we rapidly screen for those become key targets for either diagnosis or therapeutic development or whatnot? So that's where PEPperPRINT came in, where we started doing a little more systematic study, where we have a number of breast cancer tissue. So this is a study that was published 2 years ago in Journal Proteomics, so you can find the details in the paper. So what we did was we took 15 breast cancer patient samples first, And we selected, based on our pick, 348 high-frequency mutant genes. And we did an exome capture. We sequenced it high-depth sequencing. The reason for high-depth sequencing is that you can imagine tumor cells are mixed with normal cells, as well as fibroblasts or immune cells, as well as endothelial cells from blood vessels. So then if you want to find mutations, if you really want to identify, you should sequence it at high depth so that you can, even if a few tumor cells are mixed in the normal rich region, you'll still find it. So we did high depth sequencing. And the idea is that once we find all the mutations, we would send it to PEPperPRINT to design our peptide microarrays based on each patient's mutation profile. And we would get the blood samples from the same patient, and we would use the sera, which has antibodies, and incubate with the PEPperPRINT microarray to find out which of these mutations actually can be seen by the immune system and mount an effective antibody-mediated immune response. So that's how we started with the PEPperPRINT microarray. And as you can see in the next slide, here's the sequence example of high-depth sequencing, where we're showing BRCA1 gene and TP53 gene. So our exome capture was able to get about 1,300 or so reads from BRCA1 of this codon. Codon serine 509 is changed to glycine. So similarly, all the other mutations, we were getting very high read depths. For example, P53 read depths are not as high, exome capture is not as efficient, but we were getting anywhere between 900 or so to 400. So typically this kind of read depth is considered very high. So we were able to identify a number of mutations with high confidence. So we designed 15 amino acid peptides. We made a list for each patient, and we sent it to Germany to design what we call personalized peptide microarrays. A few weeks later, we got the peptide microarray. Here's the design of what we call version 1 chip. So each version 1 personalized peptide microarray has 15 amino acid sequences. We have variable 460 to 746 mutations, depending on how many mutations we found in each cancer patient. Typically, we would design 2 wild-type peptides adjacent to 2 mutant peptides. We have tested a number of possibilities. Obviously, you want to try and fit as many peptides as possible per chip, because these chips can be very useful, but they are not cheap. They are very well designed, and they're somewhat expensive for us, for a small academic lab. So what we do is we've tested 2 spots versus 1 spot versus 8 spot versus 10 or higher. And what we found was 2 spots was very reliable. And you can almost, and most of the time, we didn't find any case where, those 2 variability is too much. And it's very reliable to identify differences between wild type and mutant. So this is a chip design where we have anywhere from 460 to 746 unique wild type and mutant pairs. And then we put in what we call common breast cancer mutations, which we have mined from the literature. Now, these are the genes that are not necessarily mutated, but we wanted to know whether or not these can be immune reaction against these can be detected in the patient samples as well. So the design of the chip is that each slide can harbor 2 duplicate chips, and we can use twice or 2 different times with different amount of sera. And of course, PEPperPRINT usually have a standard either anti-FLEC or anti-HA peptides. Now they have changed it to a number of other standard peptides to mark the end of the PEPperPRINT microarray. So here's the peptide microarray design. And here's the actual data that we found from one patient sample. So this is how it looks, you know. As you can see there are 5, here in this case, we are showing 5 different mutant-specific immune reactivities. And so you see 2 wild-type peptides are not reactive and 2 mutant peptides are reactive. And you know, so these are NCOL, BRCA1, which is a well-known breast cancer gene, obscurin, mucin, and two peptides from the mucin were detected from this assay. And overall, you know, we're very happy with the PEPperPRINT microarray. We see consistent binding patterns, and we could reproduce it from chip to chip, and variability is somewhat low. So for this sample, we were able to find 5, and a number of other samples have few mutant-specific peptides that showed up. So then the next step for us was, well, you know, out of 348 genes, we were able to find very few mutation-specific signatures. So is that all that's happening? You know, granted, we were just checking only 348 genes. There are 25,000 genes that are available in human genome. So one idea is to increase that. Second idea was, well, can we test for presence of mutation signatures against other breast cancer mutated genes? You know, so there are 2 studies that we relied on, where they've looked at mutations, one from 15 breast cancer and another one from 70 breast cancer. We downloaded the mutation signatures and what we call version 2 and version 3 peptide microarray. And then in addition, we decided to mine Cosmic database, which has compiled over 9,000 cancer genomes at that time. And we downloaded most frequently mutated cancer genes and what we call version 4 diagnostic array, which is essentially looking for highest frequency mutations that are present in there. And we have a criteria for a mutant site has to be at least 17 times mutated to reduce the background. And then each of the nucleotide change has to be at least 10 times and things like that. That was determined upon looking at the background frequency. So we tested this version 2, version 3, and version 4, and we were able to identify additional signals that are present in these breast cancer patients that we have. Now, as I was mentioning, you know, although these results were positive, not as satisfactory because we were not sampling the whole genome of the patient. So then, as the last step, what we did was we sequenced the whole exomes, 25,000 genes or so, by exome capture and deep sequencing of 3 patient samples and adjacent normal regions. And then we looked at only the mutation sites, and then we designed 3 separate personalized peptide microarrays. So one version 5, what we call version 5 for patient 1 is 585 unique wild type and mutant pairs. And then we also put in a cosmic deletion, insertion, and single nucleotide variants. So a total of 8,908. So these are in duplicates. Sorry. And similarly, patient number 2 has distinct numbers. Patient number 3 has a lot of mutations, so it's 3,775 unique mutant wild type pairs. So then we had to design 2 chips for that patient. So we designed those, we tested the sera, And the results are shown here, where when we looked at, first 348 genes, we were able to find 5 tumor, what we call tumor antigens, that are mutations that invoke immune response in these patients. Whereas when we did the whole exome, we were able to find 15. Patient 2-0, if we looked at 348 genes, whole exome, we were able to find 12 tumor antigens. And patient 6-1 in 348 genes, and then whole exome allowed us to identify 8 tumor antigens. So for us, this was kind of rewarding that, if we were to look at the whole exome, and you find hundreds or sometimes thousands of mutations, whereas if you send these mutants peptides to PEPperPRINT and design personalized peptide microarray, and then you would test which of those can invoke immune response in these same patients, you can narrow down potentially things that are likely candidates for tumor vaccines or things of that nature. So here's another example where, we're comparing here patient 6 and patient 1. This is a deletion that result in frame shift. So oftentimes whenever you have deletion, what happens is frame shifts occur, and you could generate many potentially 15 amino acid peptides. So we design multiple peptides. Usually our peptides are 5 amino acid overlap. What we call deletion 1, deletion 2, and deletion 3 has 5 amino acid common, PVHDA for wild type here. And then mutant will have, mutant frame shifted right here because of deletion. So these red letters would indicate the frame shifted amino acids. So that'd be 5 amino acid overlap. And you can see that patient 1 showed immune response against deletion 1 and deletion 2 and deletion 3. Whereas another patient just showed immune response against deletion 1, but not 2 and 3. So even in those kind of cases, there's a clear patient-to-patient variability exists, which we were able to document, which is not necessarily surprising because depending on protein abundance or depending on the presence of different HLA that can bind to different peptides or many other factors, you can imagine there'll be a significant patient-to-patient variability. The key point for us here is that just because you see genomic mutation, you don't really know what's happening at the immunological level. And looking at immunological level using this kind of peptide microarray provides you with another dimension of what's happening in the patient. Here's a summary of some of these peptides that we've identified. You can see a number of, TP53, which is a well-known tumor oncogene. So there's some of them immune responses really high, some of them are not as high, right? So, and you see mutants, you see a number of other BRCA1, CDKN2, CHEK2, and a number of other proteins that are present. In that paper, we reported 149 putative tumor antigen in that paper that can be potentially be useful for downstream studies. So that's essentially the recap of the paper, and we see I'd like to spend a little bit of time on future directions and some of the work that we're doing. We see a lot of utility in peptide microarrays, the reason being that you can rapidly translate genomic sequencing into immunological readout. So it's, genomic sequencing provides you lots of data, and from there you can kind of narrow down on what are significant targets that you can use. And some of the other things that we're doing, which I touched on early on, that is still unpublished, so we can't really show it. But part of the things that we're doing is these tumor infiltrated immune cells. So you do see a lot of T cells and B cells infiltrating tumors. And these are the cells that are coming in and trying to attack or trying to sense the tumor antigen and invoking immune response. And a lot of times, there's lots and lots of information in these immune cells. So what we do typically is we look at these tumor infiltrating T cells and B cells, and we can molecularly profile antibodies, for example, and rapidly clone them, and then use PEPperPRINT Peptide Microarray to map what are the targets. As you all know, nowadays, people are really interested in finding additional tumor targets, additional antibodies that might interact with tumors for targeting using T cells or CAR T kind of approaches, or things or direct antibody therapeutics kind of approaches. So we think having PEPperPRINT kind of microarray is very helpful in that regard, especially if you want to identify a lot of solid tumors are typically not very responsive to immunotherapy, and that's a reason. Is one of the key reasons is identification of tumor antigens are not easily accomplished. So we think PEPperPRINT kind of peptide microarray has a role in this kind of space. Additionally, we're interested in developing potentially a diagnostic chip where patients can be monitored. For example, we discussed that in the paper. Essentially, we think these kind of commonly mutated genes could provide a sense of how your immune system is working, and especially patients undergoing immunotherapy such as checkpoint inhibitors, whether or not they're responding or not. You can monitor details of antibody response in patients' blood. So that's some of the things that we're interested in. And the final ultimate goal, of course, is a patient walks into the clinic, you want to, as quickly as possible, take the tumor out, molecularly profile, and come up with therapeutics that can help the patient without taking too much time. It could be a design of vaccines, for example. It could be design of antibodies, in other examples, or generating CAR T kind of vectors using these kind of antibodies. Those are some of the things that we are excited about. Essentially, that's the end of my presentation, and I'll be happy to answer questions. I want to give some conclusions, which is peptide microarrays can integrate data from proteomics, genomics, and systems biology approaches, especially system biology approaches that involve immunology, and it can help identify antigens, antibodies directly from cancer tissues. I've shown some data, hopefully to convince you that that can be done. And then we believe that insights into diagnostics and vaccines and therapies can be gained by these kind of approaches. And ultimately, we believe that these kind of pathological tissues, such as cancer tissues, has tremendous amount of information that can be harnessed for, not just for personal patient, but these kind of therapies, such as antibodies or vaccines, can be used for other patients that have similar mutations as well. So we believe harnessing a lot of information from pathological samples, and we kind of imagine future cancer therapies would be, using lots of antibodies and matching with different patients to see which patient will respond to which antibodies or vaccines, for example. With that, I'd like to give credit to people who have actually done the work. Debbie Lundgren was our bioinformatics person. Benetta Chandro is a graduate student. She's getting a PhD. Sana Aristrahova was a technician with me. She's left, but she contributed in the early work with the PEPperPRINT microarray. And we collaborate with a number of pathologists such as Purnima Haggerty at UConn Health Center. She's a breast cancer pathologist, Melinda Sanders, Christina Stevenson, a breast cancer surgeon. And we have a tissue bank, which is run by Lori Perpetua. So we have a lot of tissue, serum, and patient data that we can really harness. And thank you for your attention, and I'll be happy to take some questions.